Jerusalema ikhaya lami, ilondoloze uhambe nami, zungangishilana. Jerusalema ikhaya lami, ilondoloze uhambe nami, zungangishilana. Dawo yami ayikholana, buso wami awukholana. Ilondoloze zuhambe nami, dawo yami ayikholana, buso wami awukholana. Ilondoloze dawo yami ayikholana, buso wami awukholana.
This is fine. Okay. I sound better. Okay. Ladies and gentlemen, welcome, welcome. Good evening, good morning, good afternoon, wherever you're watching us live from. My name is Patricia Nanteza, and I am Science Stories Africa. We are very excited that you're coming along with us on our second edition. This year, we are in the theater alone. It's just a few of us, and we miss you immensely. We miss your energy. We miss your vibrance, but it's okay. We are not going to cry over spilled milk. It is what it is. So this evening, we are all about science. We are celebrating African innovation. We have 4 brilliant scientists with us this evening. So those are the stories you're going to hear. There is also entertainment. So please send that link, send that YouTube link to your friends in the groups that you're in. Remind them Science Stories Africa is about to start. But before we start, I would like to say my thank yous quite early in the program. We want to thank our parent organization, the Cornell Alliance for Science. where the idea of Science Stories started several years ago. Thank you, Cornell, for having believed in our dream and for having thrown your weight financially and otherwise having thrown your weight behind us. This is the second year, and we are grateful. This year, we got an individual farmer. You know I'm always talking about farmers. This year, we got an individual funder, and he's called Ian Gazad. Ian and Amy Gazad, thank you to your family for also having believed in the dream that Africa has great science, and we need to share this science, first of all with us, but also with the world. We appreciate your support. IITA, the National Agriculture Research Organization, thank you for giving us staff time. Science Stories Africa is run by volunteers. We are employed elsewhere, but thank you very much for having allowed us, your staff, to come work with us. What else am I supposed to say? So let us start off by getting some entertainment from Davies. So Davies, if you could please come, and don't forget to share that link. Tell people, Dr. Wayengera, Pamela Paparu, Lina Tripathi, who will be joining us from Nairobi, and Dr. Kashub. They are going to talk about their innovations. Davies is coming on stage. Let us put our hands together to welcome him, and let us enjoy this evening together.
I'm Dr. Sarah Evanja, Director of the Cornell Alliance for Science. Problems like hunger, poverty, malnutrition, and slow agricultural growth are impacting people globally. According to the United Nations World Food Program, there are over 795 million undernourished people in the world today. Hunger and malnutrition are in fact the #1 risk to health worldwide, greater than AIDS, malaria, and TB combined. Recent innovations could play a critical role in helping to solve these problems, and biotechnology is at the heart of this discussion. With an ever-increasing population and continued concern about environmental degradation, the goals of farmers, scientists, environmentalists, and other stakeholders are really not that different. The stakes of these humanitarian challenges demand that we use scientific evidence as one of the guiding principles in making policy decisions that are necessary to make innovations accessible to the people who need them most. Advances in research and access to new technologies have the potential to help provide a sustainable and safe food source for the world. We're currently seeing public sector scientists using plant biotechnologies to address these challenges, whether in combating banana bacterial wilt disease in Uganda, fruit and shoot borer damage of eggplant in Bangladesh, or even the ring spot virus's impact on the Hawaiian papaya. Based at Cornell University in Ithaca, New York, the Alliance for Science is a communications-based, nonprofit organization represented by scientists, farmers, NGOs, journalists, and concerned citizens from around the world who have come together to tell their stories through the use of interactive online platforms, multimedia resources, and communication training programs. Ultimately, we seek to restore the importance of scientific evidence in decision-making. We use data to make informed choices about the use of biotechnology as a tool for advancing food security, environmental protection, and health. We hope to empower our allies at the grassroots level, building a global movement to advocate for access to innovations that can play an important role in addressing these challenges. And we invite you to join us in this cause. Become an ally of the Alliance for Science and make a difference in the world today.
So, ladies and gentlemen, good evening yet again. Sarah has made a very good call. Become an ally of the Cornell Alliance for Science. And when you do that, you become an ally for science, and with science, improving people's livelihoods and ensuring that science does what it's meant to do, that is solve the problems, people's problems, both in Africa and elsewhere. So go to our website, Cornell Alliance for Science, click join, and that's enough. You would have become a member. But also, Science Stories Africa, please join us, follow us on Twitter, on Facebook. If you write emails, write us an email and tell us about the people in your community who are doing exciting work that you would want us to share their stories on our platform. Coming up next, our very first speaker, ladies and gentlemen. He is an immensely brilliant scientist. When we were having our training, I was even scared. I was like, now how am I going to introduce this man? I'm in the presence of research royalty, academic royalty. Dr. Misach Wayengera is the brains behind the Ebola testing, the Ebola diagnostic kit, which is really a kit that is used to test Ebola. He tells us that this kit is like a pregnancy test or a paper strip. It gives you results in just 2 to 3 minutes. This has a lot of implications in the medical world, because if you cannot diagnose a disease, you cannot heal it. We have seen this with COVID. So diagnosis is very important. It's the very beginning point in curing whatever disease you're suffering from. So ladies and gentlemen, welcome with me one of Africa's most brilliant brains. We are absolutely honored and privileged to share with you our platform, Dr. Wayengera. So as he comes up, please put your hands together. I'll just put the mic.
Thank you, Patricia, for the introductions. Good evening, viewers. August 2019, and I'm in Congo, Brazzaville, at the headquarters of the WHO Afro-regional offices. I am here to receive WHO's Medal of Honor and recognition that is awarded to young African innovators for health. There are moments in life that are essentially exhilarating in terms of emotions. And this is one of the moments that I've experienced. You imagine me standing before the director of WHO at the global level, Dr. Tedros, and right in the midst is Dr. Moiti, who is the director for the Africa regional offices. These moments, of course, demand excitement, but occasionally, what happens is that you find yourself essentially humbled. And the reason for the humbling is not because it is not a moment of excitement, but the fact that a dream that you've been pursuing for so many years is finally coming true. That is amazing. And I'm here to receive the prize because of our relentless efforts over a span of close to 20 years to develop a rapid diagnostic test for Ebola and Marburg. Let me tell you, why is this relevant, the story that I'm telling now? Ebola and Marburg are literally generic members or sisters of the same family of viruses that we call the Filoviridae family of viruses. These viruses cause rare but highly fatal hemorrhagic fevers within remote equatorial African villages. Occasionally, we see these viral hemorrhagic fevers spread to the urban centers. And as we saw in the West African outbreak of 2014 through 2016, the pathogens have potential to cause public health emergencies of international concern. While these diseases are easy to prevent, when you are able to diagnose the disease right and isolate the infected people, the existing technology platforms for diagnosing viral hemorrhagic fevers that are caused by Ebola and Marburg are not suited for deployment at the point of care within the remote equatorial African villages, occasionally where there are no laboratories, there are no experts there. Now, here's the importance of what we are doing, or what we did, what we have done. The test we are talking about is a fast test that yields results within 2 to 3 minutes. It is easy to use, doesn't require a lot of expertise, literally designed on the platform of what we call the lateral flow tests or the paper strip tests or the litmus tests, which literally resembles the pregnancy test. When a woman who is pregnant goes to visit the doctor and says, oh, I'm pregnant, I suspect I could be pregnant, I've missed my periods, I want to find out if I'm pregnant, the doctor simply hands over to her a test strip and gives her instructions, a few instructions on what to do, and she will go into the laboratory, put on the sample, and she's able to tell whether she's pregnant or not. And this is what we are talking about. But most importantly is that we're talking about a test that is affordable within the setting of sub-Saharan Africa or equatorial Africa. A test that costs within under $3. Ladies and gentlemen, how did we come to this? In the year 2000, and this is the year I was actually in my second year of medical school, Uganda experienced what was then the biggest Ebola outbreak. Over 570 people got infected, and of these, because of the high fatality rates that are associated with Ebola, we lost 270 lives. Among the lives included the lives of Dr. Matthew Lukwia, alongside several nurses. But here's the trick. The pandemic caused social disruption in the districts of Masindi and Gulu, where the cases were very heavy, including Kampala. And yet, at the time, the best we could do was to pick the samples, move them to Uganda Virus Institute, and ship them to CDC Atlanta. You can picture it would take us a week or 2 weeks to get results. For Ebola, even a day is too long to wait, especially when the patients have progressed to the prodrome stage. These experiences, alongside many others, treating patients with HIV, and of course realizing the vulnerability that Africa has in terms of, having to wait on the West for solutions, made me rethink the reason I went to medical school. When I've completed my residency in 2005, I worked briefly as a coordinator for a research program run by Makerere University. In 2006, I was lucky to receive a fellowship to do a program in bioentrepreneurship at the University of Toronto Medical and Discovery Research District. That's MassDD. I easily took up this, knowing very well that this was the path to empower me to begin working towards solutions that will help Africans. During this program, my research focus was literally studying new modalities for gene therapy against HIV. We were identifying enzymes in bacteria that literally are able to cut up the HIV genome, destroy it, with a focus of developing them as gene therapeutics. In the process, I acquired skills that could turn out to be of relevance across pathogens. This is what we call bioinformatics. Bioinformatics is literally the utilization of computational agility to analyze a lot of data in a very quick span. In the process, when I was trying to find strategies of identifying these enzymes, of course, I sought out partnerships across North America, and we finally came across platforms that had already been developed by industry that were producing these enzymes in terms of identifying the targets. And through the process, we screened over 2,000 enzymes. We were able, of course, to identify several of them that could cut up the HIV virus. And that work is published in the African Journal of Biotechnology. But that's just the start of the story. Once I acquired this technology, began accessing global databases for pathogens, including the NCBI, the National Center for Biotechnology, which is hosted in the US. I realized we have a lot of information out there already, sequenced pathogens, Plasmodium, the mosquitoes themselves that spread malaria, Mycobacterium, the cause for TB, Ebola and Marburg, the viruses. I rapidly moved into the process of literally studying each of these pathogens. That's when we started to study the genomes, or what we call the entire genetic makeup of Ebola and Marburg. At the same time, we studied the protein makeup of these viruses. And of particular interest, we picked interest in a protein which is on the surface, what we call the spike proteins. These are more like the hairs that are on the body of the virus. We began studying these proteins, comparing the protein from Ebola species to the proteins from Marburg. We found quite interesting things. The first thing we found is, obviously, because the viruses are related, they are sisters, these proteins were essentially quite very similar. But we also noticed that there were differences between the proteins of Ebola and Marburg. And the second thing, of course, we noticed, the third thing we noticed was that even within the Ebola species, you could be able to make a few differentiation. This led us to, of course, hypothesize, come to the aha insight that we could potentially use these as targets for developing easy tests that can be deployed within the setting of Equatorial Africa. Now, anybody who has done research or research and development, what we call product development, knows it's one thing to, have this big idea of, what you want to do. You want to go to Mars. It's another thing for you to actually meet that target. Between 2007 and 2012, we made a lot of attempts trying to apply for grants to get money, because the proof of concept experiments demand money. Validation experiments demand money. We applied to the US National Institute of Health and a program for biodefense. And what we were proposing, of course, was to build a sophisticated laboratory, but also advance these molecules. We never got funded. At that time, of course, we also reached out to the country, the leadership of the country, warning them that these pathogens could be used as pathogens of bioterror. And it was important that we develop capacity to deal with these organisms. But after some time, of course, following these failures, luck favored us. Or should it say we were prepared and, luck came our way. During this time, Grand Challenges Canada announced their seeding grants, and we applied to the seeding grants. We partially applied to the STARS in Global Health Program, and we got lucky. We were awarded a seed grant of 100,000 Canadian dollars. And the project, of course, that we had gone out, set out to do was development of a rapid diagnostic test for Ebola. But the key thing here is that we wanted to conduct proof of concept experiments, really try to find out these targets that we have identified. Can they indeed react with the Ebola samples and give us results that we can then move to develop a test? It was impressive because at everything down, we dived into this data. Lucky for us, the data was extremely positive. The peptides were actually very well reacting with the antibodies within the samples, and the polyclonals were also capturing the virus right. So we hurried this up and got the report together. Once the report was together, submitted it, and that's when we realized that actually Grand Challenges was interested in rapidly transitioning us to what the second phase of funding, from seed funding to what we call the transition to scale funding. Now, we had challenges at this point. Transition to scale funding demands that you need to have a matching funder. The funder gives you $1 million on the other side. You need to have another funder who provides another $1 million. We didn't have this money. But, you know, because of what was happening in West Africa, the peaking of the virus, the fact that the virus had begun to jump from continent to continent, it became obvious that everybody realized that if we had a simple test that could be deployed within the communities and to differentiate between the people who are infected and not infected, it would work out very easily. I will tell you, in West Africa, because I was in Sierra Leone, we believe that there are people who had diseases like malaria and ended up in the Ebola quarantine centers because we did not have a rapid test, or the platform for PCR would take several hours, and that you wouldn't allow these people to come in contact with the other community. We got, finally, of course, through the process, Grand Challenges employed an independent monitor for diagnostics, Halteas, LLC from California. They came over, they looked at our data, we made the presentations to them, they visited our labs. At that time, we had started to partner with a small company, Ugandan company that you probably don't hear about, Astel Diagnostics Uganda Limited. This company actually makes pregnancy tests, it makes malaria tests, it makes hepatitis B tests. This company is GMP certified, this company is WHO accredited. These tests are actually sold not only to the Ugandan government, they are sold literally to much of sub-Saharan Africa. We visited the facility, and of course, we passed that phase, and we were, because of the fact that we didn't have this extra matching money, Grand Challenges put down $650,000 US to start off. We used this money to produce a more refined version of the targets, what we call monoclonal antibodies. And we tested over 12 monoclonal antibodies producing over 64 sandwiches. We talk about sandwich because in order for you to produce this lateral flow test, you need an antibody that captures one side of the virus and then a second antibody that sandwiches the virus. So we produced about 12 really good working sandwiches. And we got, at this time, all along, everybody was excited. The funder was excited. The external monitors from Halteas were all excited. We were excited because we knew, oh, everything is it. We're on target. We are going to deploy the testing in West Africa. The next phase for validating this came, of course, taking it down to the P4 and testing it on live virus. We, under partnership that we have with Professor Paweska at the NICD, we were invited, we went in to work in the P4, we cultured Ebola Zaire viruses within cells, and of course, began the testing. The initial testing, nothing was working. Repeated testing, nothing is working. We went back and said, maybe there's a problem. We carried wrong antibodies. We started testing the same antibodies with the recombinant protein we had expressed in HEC cells, and they would work. But when it came to the natural virus, they failed. I'll tell you, this is one of the most frustrating moments for a scientist, when experiments don't go. Things are ringing. And then, you know, for product development, you've set milestones. The funder is looking at them. The world has expectations, because at the time we knew our product was the solution that was needed in West Africa. And it was very frustrating. Everybody looking at us and expecting what is going on. Luckily enough, of course, we had been with the monitors for quite a long time. And the funds that we had were also dwindling. They were over. It was another year of silence, everybody wondering what is happening. But I'll tell you about that 1 year. Every time I would go to sleep, my mind would keep asking, what went wrong? What is the problem? It took us a whole year to actually find out that the protein that we are targeting on the virus, the spikes, they get masked or kept, in which case they hide. In a sense, I think this is a modification the virus takes up to protect itself from direct attack. And that's when we began realizing, for this to work, we need to find a way of removing the mask or the cap. So, 2017, we applied for a European developing country clinical trial partnership with aim of trying to develop these platforms. The same year, next year, 2018, we won an emergency Ebola grant, and that was aimed at literally validating what we call the buffer system that we need to wash off. If you have used a rapid diagnostic test based on the litmus test or lateral flow test, you put the sample on the test, on what we call the sample pad, but you have a small buffer that you add on to enable the sample flow on the test. And that's what we are trying to work with. It was, I think, this start November, December, we began getting hits, 2018, of buffer systems that could actually wash out and enable the monoclonal antibodies. And at this time, of course, my graduate students who had been working with me were all excited, because it was like reawakening the dream that we had been pursuing for quite a long time. Everybody was, you know, and one of the students says, hey, but WHO, they have announced an innovation challenge, and we believe this is what you need. We're going to put in an application. It's like, fine, no problem. So they put in the application. I sent the application, and out of 400 applicants, 30 of us were chosen. And we went to Cape Verde, the 30 of us, for an interministerial meeting of the WHO Afro region. And at that time, we had to display our prototypes. And I carried my prototypes. I'm not sure whether you have anything of this, but this is a sample of what I walk around with. I was supposed to bring a bigger portion of it, but I would like you to look at this. These are the tests, and we have a sample pad testing test line and then a control line. So I went with this. Little did I know that some of the people who were there were actually people that were in the outbreak, responding to the outbreak in West Africa. And this is how we emerge as the best. This is how we get chosen as the best under the category of product development. Now let me tell you something. It is very difficult sometimes to explain emotions to other people, especially when you've been pursuing a goal for a long time. And this is why I'm telling, at the start I said, okay, the experience is exaggerating, but at the same time, of course, the relief that comes with it. Because for us, when we set out to do this, the whole world knew we are doing this, and our failures would literally bring the same frustration to the entire world. And that's why at the end of the day, the excitement for us came more as a relief, because finally we knew we have a product that will be deployed on large scale in sub-Saharan Africa. Now, the other thing I will tell you is these pathogens are classified Class A pathogens. Before we did this work, Uganda was having rampant outbreaks, and we kept saying, is somebody walking in with these pathogens trying to frustrate us? And when we talk about biodefense or biological defense, it's unimaginable. But this is the next phase of, a lot of things. And it is important that we invest. Now, everybody asks, why do we do this? I think that I set off on my path largely with the aim of inspiring other scientists, younger people. Africa lagged behind in the Industrial Revolution. At the time, we were being sold as slaves, and we can't blame it. But I'll tell you, my research focus, on the other hand, my lab, and the groups and the students I train, our focus looks at the DNA makeup of pathogens. Why? Because we know infectious diseases remain a big problem for Africa. And unless we resolve this, I'll tell you, our budgets, our health systems will not be able to deal with the emerging pandemic of non-communicable diseases. And this is why we've really spent a lot of time in this direction. If you look at my work, I will tell you this is not the first thing we are dealing with. We are developing TB diagnostics. More recently, we've gotten funding to develop COVID-19 diagnostics. We're developing an HIV therapeutic, what I've talked to you, gene therapy. But why do we do this? Because we believe it is time to build a critical mass. Does Africa have to reinvent the wheel? You keep asking. We can buy drugs from elsewhere. But the truth of the matter is, I believe that there is technology stuck in the wheel that we need to acquire if we are going to move to the next phase. Ladies and gentlemen, thank you for listening to me. I wish you a good evening.
Yes, thank you very much. Ladies and gentlemen, hope you're all enjoying the show. Thank you very much, our partners, the Cornell Alliance for Science, Ian Gazad, and IITA. Thank you very much for your support and for making this possible. So our next speaker is a lady. Being a woman scientist has very many challenges that come with family, being a mother. And now imagine those challenges are coupled with another challenge of you having to identify a new problem and presenting a solution to it. Now, ladies and gentlemen, our next speaker overcame all these challenges and was able to present farmers with beans that are resistant to the root rot disease. Ladies and gentlemen, join me in welcoming Dr. Pamela Paparou.
Hello, everyone. When did you last eat beans? I know if you are to respond to me, your answer would be today, yesterday, or a few days back. Beans are eaten almost daily by Ugandan households. However, bean production is constrained by several factors, and among these are bean root rots, which are currently one of the biggest diseases of beans. Just imagine this. A farmer in your village has planted an acre of beans, and she's expecting to harvest 10 bags. But then, her garden is hit by bean root rots. She only harvests 3 bags. That is how devastating the bean root rots can be. In 2008, I got a job as the bean doctor at the National Agricultural Research Organization in Namlonge. I was excited because I knew I was going to practice science after my PhD. However, my excitement did not last long because I soon realized I had no resources to start any activities. I soon started to wonder if I was in the right place. And this went on for 3 years. But I was determined to make this change. So in 2011, a colleague walked into my office with a call for grant proposals from Bill and Melinda Gates Foundation, DFID, and the British Biological Sciences Research Council. And this call was targeting young African scientists that had novel ideas that would solve a problem faced by smallholder farmers in Africa. After looking at this call, I realized I was perfect for this call. And after all, I had a novel idea. In an earlier survey in 2009 and 2010, we had observed in farmers' fields that beans were wilting. So many beans were wilting, drying and dying away, leaving patches in the garden. And this disease was what I would say beautiful because it had this white mycelia, silky, something like cotton, and something like beads, brown and beautiful. But then, that is dangerous because when rain comes, these structures are carried to the next field, from one field to another. So this disease was spreading from garden to garden. Then I realized this is the problem I am going to solve. However, at that time, I had a condition. I was 8 months pregnant. And for mothers who are watching me right now, you know what it means to be 8 months pregnant. But I was not going to let this stop me. So I quickly put on my resilience shoe, which I think is always very close to me. I marched forwards, never to look back. So I put together a team of scientists that I was going to work with, and we started writing the proposal. In April, we submitted the first document of the proposal, for the proposal was in 2 phases. And the following month, which was May, I went and had my baby. And this baby, we named him Jonathan. 5 weeks after Jonathan was delivered, as I was enjoying motherhood with no worries at all, I received communication from the donors that our proposal was successful. However, now the project leader had to travel to UK to be trained in how to develop the next stage of the proposal. I was very excited. I screamed because that was the news I had always wanted. And I knew I was now closer to my dream of getting a grant to do what I've always wanted to do. The kids wondered what was wrong with me, but I told them there's nothing wrong. I'm only happy. But suddenly, I looked at sweet little Jonathan. I'm like, how am I going to even go with this baby to UK, only 5 weeks old, and I had never been to UK. I'd been to South Africa, the farthest. So my heart sank. But after talking with my husband, we agreed that I could go with Jonathan. So I was excited that he had accepted for me to travel with the baby. So I quickly sent an email to the donors saying that I would like to come, but this is my condition. I have a baby whom I cannot leave behind. I must come with the baby. Then their reply was, unfortunately, we don't have a policy that supports mothers traveling with babies. That news was very devastating to me. After my husband saw how distressed I was, for he had really known how much this opportunity meant to me, he decided, I am going to finance your travel with Jonathan to the UK. So you informed organizers that you can take care of Jonathan. I was very happy because my husband has always been very supportive of my career. So I quickly sent an email again telling them, now I am able to take care of Jonathan's course and we are coming. So I think my determination moved the organizers so much that the following day I received an email. Surprisingly, I wasn't expecting it, saying that we are going to cover all costs associated with Jonathan's travel to UK, and we are even going to get a nanny to look after Jonathan as you do the training. That was unbelievable because I didn't expect that from the first response. So, we traveled and we arrived at London Heathrow. My challenges were not yet over. My first time in London, I was met with very long queues at the immigration. Then I was met with very fast-moving trains that didn't care whether you had a baby, luggage, or whatever. Then the immigration officers were asking questions that would even make me feel that this is not my baby. And the baby was so tired and irritable because of 8 hours of flight from Uganda to UK. However, we managed and we reached the hotel. So the training started. I still had issues. I was not trusting this nanny they gave me. I kept moving from the conference room to my hotel room because the nanny was sitting with the baby in the hotel room. So I kept moving up and down. I couldn't settle. I'm like, what if this lady goes away with my baby? But somehow I settled. I finished the week of the training. And then we had 2 more days to go meet our partners. At that time, my partner was in Cambridge University, the one you know. And we had to travel. Again, we moved by train. It was not easy. But fortunately for me, after arriving at Cambridge, I met a professor. I had never seen him before. A professor who was very supportive and understanding. He took Jonathan and I into his office, gave us a corner where we would put Jonathan and work on the proposal. Every now and then he would carry Jonathan if he cried while I was busy on the computer. That was simply amazing. After 10 days of adventure in the UK, we returned to Uganda. And I must tell you, this trip changed my life forever. And I'm not afraid to say that. If I had not taken this decision to go with Jonathan, I could never have been where I am today. So we returned. I continued to work on the proposal and submitted it. In July 2012 now, proposals take time to get approved. I received communication that our grant was successful. And I must tell you, among 10 grants, only ours was successful. I was very excited. However, that was short-lived because I realized now we had to start doing what we had promised to do. The first thing we had to do then was to validate what we had claimed. We said the bean root rot is a major disease of beans in Uganda and is rampant, but we had just said that. So we needed to now validate that. So what we did was to carry out a survey in bean growing regions of Uganda. We went to almost 3/4 of the districts of Uganda, and indeed we confirmed that this disease was very rampant and farmers were suffering. But farmers had nothing to do about this disease still, as we had observed earlier. Then we returned to Namlonge after the surveys, and we set out to do what we had promised to do in our proposal, which is develop a bean that will be resistant to the root rot. And readers out there know that this follows a well-designed set of protocols. After the surveys, we then had to characterize the pathogen. You can liken this to how people were talking about the COVID, the virus, that the Ugandan strain is not strong. That's why we were not dying in the beginning. The Kenyan strain is stronger. So we needed to know that about our organism. So we collected several of them and we characterized them to know which ones are really strong and which ones are not. And then from there, we needed to screen as many bean varieties as we can to identify if only even one variety that would be resistant to Sclerosium root rot. So while doing this, it took us almost 2 years. It's not an easy task. We collected over 500, actually more than 500 beans, most of them internationally through a research NGO in Kawanda, but we also have local bean varieties that we used. So we started screening these beans in the screen house at Namlonge, inoculating each one of them with Sclerosium pathogen. We used tons and tons of soil, hundreds of trays, and each of these trays is 20 kilograms. This was not easy. And we physically evaluated one by one thousands of plants. But this was not giving us what we wanted. As the project leader, I had in my mind a picture, a wonderful picture of what I wanted to see, which was I would walk into my screen house and look at trays. We always had over 35 at a go, 35 to 50. I would see these trays, and in them, there would be wilting plants. Maybe you have seen some in the pictures behind me. Several of them wilting, but then you would see this one variety that would be standing and growing while others fell off. But this image was not coming. For almost 2 years, we almost gave up. And if we didn't see this image, that means our project would have ended there. So this was not a nice thing for a scientist, things not working out. But fortunately for us, in one of the experiments we set up towards the end, there were 3 varieties. We didn't know how they would react, but we planted them. So we started our evaluations, germination. These varieties germinated so well. Out of 20 planted, 20 germinated. And this was very rare with this disease. So we were excited. Something was beginning to happen. So we kept going in the screen house, even sometimes twice daily, because we had to make sure that things were not turning the other way. So these beans started, they were growing well, they kept growing, and our excitement was growing as we took pictures over pictures because the image we wanted was now coming up. And luckily for us, on the 28th day of planting, which normally would be the last day of assessment, these plants were still doing as we expected. More than 90% of them had not died of this disease, and they were still standing. This was nice, but was it? For science, you cannot celebrate yet. It might have been chance. So we needed to confirm, indeed, what we are seeing is what we want to see. So we repeatedly planted these varieties and infected them, but they still behaved the same. And finally, it dawned on us that we had found what we were looking for. We were very excited. We had done it. For the first time, we had identified 3 bean varieties that were resistant to Sclerotium root rot. As I said, you achieve one, you think of the next. Then we realized, oh, but these varieties, if we send them to the farmers like this, they will never take them because they are small-seeded and they are red in color. The Ugandan consumer wants their bean yellow, mostly large-seeded, yellow, speckled, the one they call nuds. I'm sure you've seen that in the markets. And then they want it white, they want it black, but these beans were not. So then we needed to do something to transfer this good character we had seen in this not much liked bean into our liked bean. And we knew we could do this through plant breeding. So we set out, the team at Namlonge. I'm not a breeder, we have a breeder. So they made their crosses and they gave those offsprings, the babies of that cross, to me to evaluate. That's how you get new varieties. You cross them and then you get offsprings and you develop them into what you would like. So they gave me lines, several of them. We screened them the same way as I told you earlier, planted them, infected them, and evaluated them. And I'm very happy to say that of these, currently, we have stored over 80 lines that we think can be used in efforts to continue with the Sclerotium root rot breeding work. And these are stored at NACRI and SIAT. So, we hadn't yet developed a variety to take to the farmers, of course. We had found a resistance, but no variety. But funds were over. The project ended. After some time, late last year, I received communication from the funder that actually they were asking me, are you interested in continuing with the work to develop beans resistant to Sclerotium root rot? I read this email, I read it again, I don't know how many times I read it. I even gave it to my colleague, please, is this true? So he told me, yes, I think they want to give you money to continue with your work. I was very excited. In my response, I said, yes, I would like to continue with this work. So right now we're in the process of granting. But at the end of our project, we shall have developed novel resources never developed before for Sclerotium root rot breeding. And these will be useful not only in Uganda, but all bean growing regions of the world. Secondly, we shall have developed a variety that is resistant to Sclerotium root rot. And hopefully, our farmers will be able to adopt this and grow it. Last but not least, we shall have trained a Ugandan scientist in next generation breeding technologies that are currently, I mean, very important if you must get varieties very fast. Thank you.
It is time to give the world's least loved hero a chance. I've got my cape on, tights too, and I'm ready to save lives. But every time I leave the house, some people scream, "Witch!" Maybe it is my superpowers. They scare some people. My talents could do wonderful things for people if they had just let me. I could save crops from dying. I could save children from starving. My friends, coffee and electricity, say they had witch to at first. They say I just have to wait. And one day, people will love me. I do not want people to love me blindly, but I wish they would look at who I really am and what I can do. They will see that I am really not that scary after all. They might even love me. I am genetic engineering, also known as GMO.
Thank you, the GMO sheroes, that is Jordana and Joanna. You've told us a good poem that encourages us to, You've told us a good poem that encourages us to assess the opportunity that comes with the genetic engineering technology. We are lucky that in our lifetime, we have seen a technology that can address most of the concerns that were otherwise thought to be unsolvable. And we wish that people would examine the technology for what it is actually and look at the evidence and the opportunity it presents us. It could be in health, it could be in agriculture, but biotech presents us with a very strong opportunity to address most of the concerns that were hitherto not possible to be solved. Speaking of biotechnology, our next speaker comes to us live from Nairobi, IIT Nairobi, and she's going to tell us about her journey because she pioneered the use of genetic editing, the use of biotechnology in solving most of the major diseases in bananas. Please welcome with me, Dr. Leena Tripathi.
Hello everyone, I'm Leena Tripathi, Principal Scientist working at IITA, International Institute of Tropical Agriculture, based in Nairobi, Kenya. I'm also the Deputy Regional Director for Eastern Africa Hub of IITA. So today I'm going to tell you my story of establishing the genome editing in Nairobi, Kenya. So let me take you a few years back into my journey. 12th July 2004. It was an early morning of a pleasant day in Kampala, Uganda. There was a slight early morning breeze as sun coming up in the horizon. I woke up early and got ready and drove to Kawanda to pick my colleague. We were actually planning to visit a farmer in a village near Gayaza. So we were on the road for almost an hour, and then soon we entered into a bumpy Marham road. I looked out from the window of my car and the reality struck me. So before actually coming to the village, I already knew about a problem which the farmers were facing in their banana cultivation. So when I saw out of the window of my car, I saw the wilted banana plants with some barren field. So we parked our car, my colleagues and I, we got out of the car and we looked around. the view was heartbreaking, because as far as our eyes can go, we only saw the diseased banana plants wilted, and the whole field was like wiped off. And in the corner of that field, we saw a small house. It was a thatched roof house. That was our farmer's house. So we entered into the house, and the farmer was there. He was sitting at the corner of the room. He looked at us with very sad, dull, and depressed eyes. So when we went with a small gift, a bag of sugar for him, and I presented this gift to that farmer. The farmer was almost in tears, and he said, he didn't have even sugar to prepare his tea. So then the farmer stood up, took us out into his field, and he started telling us how his farm was devastated because of this deadly disease. And while he was narrating his story, he was in tears. And after hearing his story, I was in tears. And I had so much of mixed feeling, which I cannot explain in words, but I was very, very sad. On other hand, there was also the desire developed in me to help this farmer. This was a very old farmer, and even, his clothes were very old and teared off, So he explained to us that this field was his sole way of getting food and income to buy things like sugar. So because the field is not there, he had no income. This was not the only farmer who was affected. There were millions of farmers who depend upon banana for their food and livelihood were devastated. So banana xanthomonas wilt, that was the disease which was affecting the farms of the farmers in that village, and also not only in Uganda but also in the neighboring countries. So this disease is a bacterial disease which affects all different kinds of bananas, and it starts with the wilting of the disease and eventually killing the whole plant. The fruits get premature ripe and rotten. So in the end, the farmers cannot get any harvest. Even some of the fruits which looks okay, but if you break those fruits, you will see that they are rotten from inside, so they are uneatable. So that's not it. The disease is spread very fast. So once one plant is sick in that field, actually the whole field will be wiped off very quickly within a short period of time. This happens because the disease is spread through the insects which are there in the field already, but also through the infected gardening tool and the infected planting material. Once the disease is in the field, there is no other remedy than to cut off the diseased plants and leave the field fallow for some time before the farmers can plant again. So something needs to be done. There was estimated economic losses about $2 to $8 billion US in 1 decade. So you can see how big is the impact of this disease. Not only the farmers who are affected with this disease, there are also the consumers who are getting affected with this disease, because the prices of banana is going up because the production is going down. So you might be wondering, what's my role in it? So my team and I, we started developing the solution to control this disease. And at IITA, we were working very closely with NARO, National Agricultural Research Organization, at Kawanda, and we were busy developing the bananas resistant to bacterial disease by bringing the disease resistance gene from sweet pepper to banana. So we tested these improved varieties in the greenhouse, and we got very promising result. We were very excited. Then we tested in between 2010 to 2012, we tested these plants under the confined field trial at the Narrows field in Kawanda. And we got very high, enhanced disease resistance under the field condition. So we were very, very excited about that. But it will still take some time before the farmers can get these varieties, because these are the transgenic bananas and you need to follow the national regulatory guidelines before these varieties can be released to the farmer. Then another phase of my life came in, and I was relocated to Nairobi, Kenya in 2011. So I was a bit unhappy to leave Uganda, but my family and I, we packed the bags and we moved to Nairobi. I was a bit sad, but because I have positive attitude, so I also was a bit excited that, okay, so this is an opportunity to get more excitements, new memories in a new country. So after coming to Kenya, my team and me, we created a plant transformation platform. It's only a type in Africa. This is the only one platform in Africa which can be used for developing the genome edited banana, but even the other crops in sub-Saharan Africa. And once we started working on this, I was full of joy. And then this was actually, I will say, the most excitement in my life so far. So what is genome editing? Genome editing is a group of technologies which allows scientists to make precise and specific changes in the characteristic of an organism. These type of tools can be used for crop improvement, for example, for making bananas resistant to diseases and pests, and even increasing their yield. So genome editing is not something new. It has been there since ages. Nature has been doing the editing of crops since ages. So in olden age, actually the nature was changing the crop varieties, and then farmers were selecting them based on their needs. But in early 20s, radiation and chemicals were used to speed up making these changes, but still the changes were random. Later on, some other technologies were advanced and to make these changes precise. And one of the technology which was recently developed was CRISPR-Cas9. So two women scientists, Jennifer Doudna and Emmanuel Carpentier, won the Nobel Prize for this technology this year for their CRISPR work. So CRISPR-Cas9 is available naturally as an immune system in bacteria against the invading viruses. So this is also known as molecular scissor, which makes a cut into the DNA of a cell and then gets repaired using the cell's own machinery without bringing any foreign gene. In 2013, CRISPR-Cas9 technology was customized for its application in plants and animals. So one day in early 2014, I was sitting in my office reading an article on application of CRISPR-Cas9 for plant improvement. And I wondered, can I use this technology for improvement of banana, for making some small changes in banana to make it disease and pest resistance? And as I was going through the article, I was a bit nervous, but also very excited because all these different type of thinking was coming in my mind. So I was a bit nervous because in those days, the genome editing was very much limited actually to the advanced labs in the developed countries, and there was no single report in Africa. So I wondered, why is it not in Africa? Whether there are no resources in Africa, or there is no capacity or training. And I took it as a challenge and made very strong determination that I'm going to establish genome editing platform for the crop improvement in Kenya. And there were many things actually which encouraged me to think in that direction, because we already have the tissue culture transformation platform available. The lab facilities were there. The protocols required for making the editing in bananas were there. And then the reference banana genome sequences were there as well. And then definitely the strong determination was there. So we started working on this. And then to start a genome editing platform, actually the target we chose was the banana streak virus, which is another devastating disease affecting bananas in Africa. BXW disease was still in my mind, but at that point I was not thinking of any strategy which can be applied for developing the bacterial disease resistance. So that's why, you know, I thought about, okay, let's work on the banana streak virus to establish the platform, and then we will use that platform for developing the BXW resistance. So in 2018, we got the breakthrough, and we deactivated the banana streak virus, which was integrated into the host genome of banana using the CRISPR-Cas9 technology. And this was the first successful application of genome editing in banana for the disease resistance. It was the first report globally for any disease resistance using the CRISPR-Cas9 in banana. And so was a happiest day of my life, and many people thought it was unbelievable. The planning for this actually started in 2014. So over the weekend, one day, I was sitting in my house reading an article. So the article I was reading was on human viruses. And after reading that article, actually, the light bulb blew in my mind. And I came up with an idea, very excited. And I went to my husband, you know, and discussed with him, because my husband has background in medical science. So I discussed with him. Later on, I discussed with the banana experts. I also discussed some of my collaborators. And then I came up with a plan after a lot of discussion and brainstorming. And then we started working on this project for deactivating the banana streak virus. And you can imagine, you can feel how excited my entire team was. Not only my team, but even the nation was very excited, because this was the first project in Kenya, in Africa, even at IITA, applying the genome editing for crop improvement. So once this protocol was established for deactivating the virus, it has overcome an hurdle which banana breeders were facing, but also was also the germplasm movement on the continent. But this breakthrough has opened up a path for us to applying this genome editing technology for other diseases as well. And whole the time I was always thinking of the banana xanthomonas wilt, which is also in short called BXW, and the old farmers suffering because of this disease. So as you remember, I said that all different types of bananas are affected by BXW. But there are some wild type of seeded bananas which cannot be consumed because they are seeded. They have, some of them has the resistance against the banana xanthomonas wilt. And I was always wondering, what is in the wild type banana which makes them resistant to the disease, and that's not present in the other bananas? So this was always puzzling me, but this mystery was solved in 2018, and we got the information that why these wild type bananas has the resistance against this disease. So in 2018, I was actually top of the world because we had this information from wild type banana which we can transfer into the cultivated varieties of banana to make them resistant, but we also now know how to edit the bananas. We have the protocol, we have everything available. So I made a plan and we started experimenting in the lab. I used to spend hours and hours in the lab with my team members, to do all these experimentation. And finally, in 2019, we generated the edited banana plants resistance to BXW disease. And we tested them in the greenhouse. As we were expecting, we got the resistance against the BXW. We demonstrated it in the greenhouse. And that was the excitement moment for everyone. We were so excited that yes, we made it. Now we are planning to actually test these edited bananas under the field condition to see if they have the same yields like the other bananas and while retaining the disease resistance. So the bananas the edited bananas look exactly the same like the non-edited bananas because we only made very precise a small change in one characteristic, that's disease resistance. We didn't touch anything else, we didn't put any foreign gene in it, and that's what the motivation for me to start the genome editing work, because in genome editing work you don't use you don't integrate any foreign gene, you only do very little specific change into the banana's own genome. So the product developed through the genome editing are not considered as genetically modified, they are considered more like the product that varieties developed through the conventional breeding. So from here we can say that yes, if you have a strong determination, dedication, and you work hard, there is nothing you cannot achieve. So this was unbelievable for us. So we have developed a platform for genome editing which is self-sufficient. It has all the technologies and tools in place, starting from developing the CRISPR-Cas9 reagents, the tissue culture, transformation to generate the genome edited plantlets, to do the molecular characterization, to detect the targeted mutations as well as the off-targeted mutations, and then finally to evaluate them for the disease resistance. So I'm so much passionate about this platform. My team has competent scientists, research associates and students, and currently we are using this platform for developing the disease resistant improved varieties of banana, even other crops, and also to train people in order to build a strong capacity in Africa. And we are very much passionate about this platform because this platform was developed for Africans, by Africans, in Africa. You might be wondering, I'm not an African, I am basically an Indian, but by heart I am African because I have spent more than 20 years in Africa. That's where I grow professionally and personally. My 2 kids were grown in Africa. In fact, my son was born in Uganda in 2007. So you can feel it. I am African actually, basically East African. So now we are waiting for the day when this variety, the genome edited BXW resistant variety, will be available to the farmer. So there can be one roadblock for releasing this variety, and that can be the availability of the National Biosafety Guideline for regulating the gene-edited product. But the good news there is that Kenya is already developing the guideline for regulating the genome-edited product, and I am working very closely with the National Biosafety Authority in their efforts in this direction. So once the gene-edited products particularly for the disease-resistant varieties of banana will be available to the farmers, millions of farmers, not only in Uganda but in other countries like Kenya, Tanzania, DR Congo, Rwanda, Burundi will be benefited, because bacterial wilt is present in all these countries. And I'm still dreaming the day when this old farmer will start growing the disease-resistant varieties. That's the time he will get enough food to eat, but also the bananas to sell in the market so that he get money to buy sugar and new clothes for himself. And that day I will say, goal achieved. So I'm waiting for that, and I will feel that day that I have made impact on somebody's life. So we are very eagerly waiting. Maybe in few years' time, like 2 to 3 years' time, these BXW resistant bananas should be in farmers' hand in Kenya and Uganda. I hope this story has encouraged you and inspired you to do something which is unbelievable. Thank you very much.
Sunday, November 22nd, 7 to 9 p.m. East African Standard Time. Science Stories Africa presents Science Goes to the Theater, the online edition, with stories from Dr. Misach Wayengira of the Ebola Diagnostic Kit, Dr. Stephen Kashoub Tumwesije, biodegradable polyphene bags from cassava peels, Dr. Lina Tripathi on genome editing, Dr. Pamela Papalu, root rot resistant beans, Dr. Frederick Kabi, anti-tick vaccine, with performances from Naava Grey, Davies the Violinist, and the GM sheroes, under the theme, The Unbelievable. Book your attendance for free at sciencestoriesafrica.com, powered by the Cornell Alliance for Science and Ian Gazad in partnership with IITA.
Kizenzi amaso owomukwano yandagekkubo simanyi kyendagano olugendo lwawala natuuka buli bwobulawo bange arazizza obuwere sula ntudde okutuusa lwolidde emmeeme nogikakkanye ntawanya olwalibeeranye ebirowoozo bimumanyi Rendi mulabako emitima girecca yananazako bw'omu nga yenze bulamu bwe gyali kimanya alibeera omubwati ng'asuubira mwoyo omu anawanirira omutima gwange uyo yoyo omwoyo gwakalasa akasufu pambatira perekera alibeera omubwati n'onyoyo mu balungi yegombesa oyo wegonjewa ampeweta muline nesumulula yansobola teryawakana ye busobozi obumpanguza embeera yonna yeyeka aliwoyo n'onyoyo obalibeeranye ebirowoozo bimumanyi lundi mulabako emitima girecca yananazako bw'omu nga yenze bulamu labe abo abancunya oba bamaze zitowererwa ebinumba naye rose bampewuse mukwano gugumidde gwa babiri tekusangibwa buli wamu omutonzi yabakonyemu nakkiriza gubeere ogw'olubeerera kimanya alibeera omubwati nga nsuubira oyo omu anawanirira omutima gwange uyo yoyo omwoyo gwakalasa kasufu pambatira perekera ontuusa alibeera omubwati n'onyoyo kimanya alibeera omubwati nga nsuubira moyomu anawanirira omutima gwange uyo yoyo omwoyo gwakalasa kasufu pambatira alibeera omubwati n'onyoyo. I want to introduce a gentleman on this next song. Give it up for Davies, everybody. The violinist. Hey, yeah. Kera kumuketsa uliwe buchanga tambuza kasobo negendereza obutagwa mu bunnya sandyagadamanye timuregwa kuba guno mukwano gunsuma gunjagaza ye kiba kizibu nnyo okuteebereza tinaye bwatyo bwawulira nje wamba yantwala yera simanyi kikyafudde abaye Sirinya Simani Subira kubana yeyo mutasi Subira kubana mulala yenna yansingira kuba omukwano gwandaga gwansusu alina engeri gyazitoya amaaso akubako limuze nenzikwamu banga ali ku muliro nga naye sijja moto nnyumira nyera to sing along. Mani chichafude alaye Honinya sirinya Tumara talaba Mani chichafude alaye Kanyane kuwa mutufu yantute Kabelono kurabala Sije Zekamwewe omutima omwoyo n'omubiri Kansule tende kanfombe kanywere nywere makazi ewoyo sirimbaye yamalawo sikisawo chichafude sirimba unyajao kumanya ya malawo kyesabo n'obuyinza eri obulamu bwange abuwamba yantwala era Thank you so much. Give it up for Davies. Thank you so much. All right, feel free to sing this one. Wanji. Let's enjoy the science stories from music. How about that? Hey, let me see. The gentleman, let's groove. Yatenkuriwala nnyo sokitya nja kubeewala abo ababadde bafubirira okukunnemesa munkwerabire bakweddize ab'ebigambo tobawulira tebamanyi gye tulaga ne gye tuva tusaanye tugume obutego mu mukwano bungi nnyo omwomwoyo nesiga nga bwenkwesiga bino byenkugamba mazima nkulayirira nkomawaggulu kuba kwatambuza obulamu bwange bwennina tekukyanonya mulala kumomwoyo ebikenewe bisse bungi zenkute naye kyendabye sikyanonyayo mulala bwenange mbanseredde the married gentlemen. You're married, right? Hey, oh, emicembi nange kuwatanya emirimu gitumbule nga naye nnemesebwa oluusi olw'omukwano banneebuuza alinaanya mukuma nenneewayo kuguma olw'obwesigwa nga bwenkwesiga bino byembagamba balayirira. Everybody, let's go. Hey. Tolina mubenzi kuyoyo tango moyo sikamo biroto mubatobe bote kacho mukwano fuma mufume onjuwala nyo 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 hey ha mulala kuma moyo ni ibisebici yeah. When you will tell a day, want you? When I get back to the day, when I get back to the day, when I get back to the day. Thank you so much. Thank you. Thank you.
Thank you very much, Naava Grey, for that wonderful performance. I enjoyed it. I hope you who are watching enjoyed it too. I also would like to thank our sponsors, Cornell Alliance for Science and Ian Gazad. We appreciate your support and believing in Science Stories Africa right from the time when the idea was just on paper. The adverse effects of polythene paper on our environment cannot be denied. If they are not piling up the landfills in Chitezi, they are clogging the channels of Nachivubo, or littering our environment and polluting our soils. Worse still, choking our livestock to death. In Uganda, the polythene bag is commonly known as kavera, and it is part of our everyday life. We use it to carry groceries from the shops and foodstuffs from the market. And for the women, on that day when we forget our umbrellas, we use it to cover our heads to protect us from the rain. Our next speaker is a processing and chemical engineering specialist. On a quest to find a lasting solution to the polythene bag, he came up with an innovation known as the biodegradable paper bag. This bag is made from cassava peels. Can you imagine a polythene bag from cassava peels that disintegrates after 3 months? To help us understand more about this journey is Dr. Kashub Stephen. Please, ladies and gentlemen, welcome with me Dr. Stephen Kashub.
Thank you, thank you. Indeed, it is a journey. It is a journey. Dr. Kashub Stephen Tumwesije. Good evening, ladies and gentlemen, girls and boys. As a child, I grew up in my father's farm, witnessing the polythene bag, popularly known as kavera in Uganda, being eaten by our cows, and they were dying. At that time, at some point we were enjoying it, because whenever the cows die, we would eat them. But of course, our parents were not very happy. So at one point, we were required to always clean the farms of the polythene bags. At that time, of course, we were a child and we didn't know why. Now, when I relocated into the city, into the capital city, now I kept on now seeing a lot of polythene bags. Now it was like an experience. I said I saw them at home, we were praying with them, but now they are in the cities, in the channels, they are on the ground, they fly around when there is wind. I said, wow, there must be a solution. Now, later on, when I was retiring from my work at Narrow Kawanda, going home, there was a lot of traffic jam, and I decided that I make a shortcut. Now, the shortcut took me through the landslide, the Chitezi landslide, and I was later told that it was a city council landslide. Now, I remember I was playing with the polythene bags. Now, I've seen heaps of bags, not only of those polythene bags, but also other wastes. And among those wastes, I saw the food wastes. There were banana wastes, there were cassava wastes. Then I said there must be a solution. Now, in 2000, you know, the polythene bag is captivating. It is so convenient that when you go to buy your goods or your food, then you carry it very easily through rain, through sunshine. You carry it and go home. And then because of that, in Uganda, and I think globally, we have developed the model of producing, using, and throwing. And then we don't reverse that situation. So it was around June 2012 that the opportunity of training came. NARO was organizing a training for scientists, and we were told that we need to do an innovative research that is going to have a sustainable impact for our country. So what came into my mind was, I was told that you need to develop a product, a food product, that is going to do that. But in my mind, I said, okay, 60% of the polythene bags carry food from groceries, from supermarkets, from all over. I said, okay, I must look for a solution for these polythene bags. Now the opportunity came, and I decided on that. But you should remember that the polythene bag, I was thinking now, the polythene bag that stays in the soil, the polythene bag that stays for over 1,000 years without decomposing, what could be the solution? Remember at home, I've looked at the safety of animals and the safety of the environment, the polythene clogging the soils. Then later in Chitezi, and now I looked at the safety of the people who are staying around. I said, now I need to address this, and I would have addressed issues of the animals, the safety of the animals, the safety of the people, and the safety of the environment. Now time came, and I was admitted at the National University of Ireland, Cork. It is based in the southern part of Ireland. It was one Sunday evening, and I was escorted by my family to the airport. Now, you know that joy that you're going out. So we kept on saying, oh, I'm going out, I'm going out. I mean, for them, they thought that was also a good thing, going out, leaving them behind. But guess what? At the airport, my son, when I entered the waiting lounge, my son started crying. Now, I thought it was the tears of joy at that time. Little did I know that he didn't expect that I was going to leave him. At that time, I think it was about 5 years. Then when I waved bye to them, then he started crying. I said, tears of joy or what? But then he was saying, my daddy, I want my daddy, don't leave me. Then I was of course not happy, but I continued and went. So I arrived in Ireland. It was a chilly evening, and then the person who was supposed to correct me was my supervisor. She came, corrected me, and took me to the university hall where I was supposed to reside. Then he told me, Kashub, you're going to sleep here. You have people in this apartment. Befriend them, and then you'll get used to Ireland. You know, it was a new environment at that time. Of course, I had traveled before, but I was looking at, now I'm going to be here for the next 4 years in this coldness. And indeed, it was terrible that night. I couldn't believe. If I was given a chance, an opportunity to come back to Uganda, I was going to come back. But while I was still there, it was a long night, and I did not sleep properly. So in the morning, my supervisor corrected me and introduced me to the Department of Chemistry, and introduced me to the staff of the Department of Chemistry, and also the students. And then he told me, and then I was given a table and a chair, and they told me, this is where you are going to start your journey of the PhD. And then she told me, Dr. Maria, my supervisor, Dr. Maria Garaga, she told me, don't fear, these people are very friendly. You are the only black, but don't worry. These people are very friendly. You know, I was looking around, I was seeing different colors. Actually, that was my first time to know there were different colors. So I started, of course, we started talking, and asked me, I said, I'm from Uganda. They said, nice, and that was all. Then that week ended. So one Monday morning, my supervisor called me and said, we need to start the work. You need to start the work because it's, you need to know whether the cassava is going to make the polythene bags. You know, my supervisor didn't know about cassava. Actually, most of them, they didn't know about cassava. They thought were vegetables. You mean the cassava, the vegetable? I said, you know, in our country, a cassava is not a vegetable, but it's part of the main what? Main dish. So he said, okay, I'm not sure whether cassava is going to make a polythene bag, but what you do, you try your best, and then once it refuses, then we are going to use other materials to make the cassava. Then, you know, my heart, now, you know, at first, I knew I was going to make it from cassava. But now, you see those words of my supervisor, then I started fearing. I said, wow, is it going to be okay? But I was determined to make cassava, to make the polythene bag from cassava. Now then, we sat down and we made a program. So once we made a program, and then I started moving. Then it was then that week ended, another Monday morning, I went to one of the supermarkets in Cork. Cork is the second city in Ireland, and in the English supermarket. The reason was, you know, I also wanted to feel big to be in the English supermarket. And when I went there, I was not expecting to get cassava, but I got cassava roots from the English supermarket. I quickly took them to the lab because I didn't want to have any decomposition along the way. Took to the lab, and from the lab, I washed that cassava, and I started my process of making the polythene bag. Now, because of course that cassava, I was also thinking that at home, really when we are going to eat cassava, we peel and eat. Now, I also peeled the usual way and threw away the peels. And then I went through the process and made the polythene bag. Now, you know, that polythene bag was soggy, had a lot of bubbles, was tearing very fast. So I was so much discouraged. I said, now I remember the words of my supervisor that if cassava fails, we're going to use other materials. But then in Uganda, back here, I had promised Naro that I'm going to make a polythene bag out of what? Out of cassava. Then, but I said, I need to make it. I didn't tell my supervisor that day. I said, let me try. The following day, I did not stop. The following day, I got the same cassava. Now I used the whole root of cassava with the peels. Now, when I went through the same process, I made the polythene bag. It was improved. I could still see the bubbles, but now it was not tearing very fast. It was not tearing very fast. And then I said, okay, now I've made it. Then I said, okay, but what is it that from the peel to the whole root, it is now better than the other one? I said, perhaps it could be the peels. So the third day, still in the morning, I did what? I got the peels, and then I also got the whole root. I wanted to compare between the root and the whole root and the peels. Now I got the peels and the whole root. I went through the same process, and I made the polythene bag. You can't imagine. The one from the peels was extremely improved. It was so improved that I started forwarding it. It was forwarding it and coming back. And then I remember one time that I threw it down to see whether it can be torn. It was not. But still, the problem of the bubbles was still there. But at least I was now happy. My heart came back thinking that now I'm going to make a polythene sheet, a polythene bag. Now then, I called my supervisor. I've made the bags, I've made the sheets, that time there were sheets. I made the polythene sheets, the Kavera sheet, but I still have a problem of what? Of the bubbles. Then she said, wow, Kashub, have you made the sheet? I said, yes, I've made the sheet. From cassava? Yes, I said from cassava. Said, let me come. She was in the office, and she ran and came to the lab. Now, in the lab, I was shocked. When she came, she was shocked. Said, Kashub, are you sure you did this material? You did this cassava? I mean, you made this polythene bag from the cassava? I said, yes. I said, what else did you add? I said, no, this is cassava. This is pure cassava. I made it. Then I told her, but I made it from the peel. It refused. I improved it by making it from the whole root. It improved. And then I improved it further by looking only at the peels. I said, wow. She was so excited. She got the, you see, she got my polythene bag. So what she did, she put her hand and here and said, Kashub, take me a photo. She gave me her phone. Now, once I took the photo, said, Kashub, let me come back. She ran to the other staff and showed them. You see, this is now a bag. This is now a sheet that I made my student. Say, which student? Kashub. Who is Kashub? Kashub comes from Uganda. You mean Uganda? Yes, that's what they said. Now, then she quickly told the technician to come and help me and show me the machine that was going to remove the bubbles. You know, that time, once we are not using a machine, you need to know how the machine works. So when the technician came, he asked me 2 questions. He said, no, 3 questions. He said, you said you are from where? I said, Uganda. Now, do you know how to present? I said, no. To make a presentation? I said, no. Do you know how to use a test tube? I said, no. Then he said, perfect. I'm going to teach you all the 2, what to do. And then Of course, there was no, making a presentation was not in the lab, but he showed me how to use a test tube. And then he also showed me the machine that I used to remove the what? The bubbles. Now I did that. I used that machine and to remove the bubbles, and I was very excited because the bubbles went, and then the thing was as smooth as you see it here. Was very smooth. And then I said, okay, I'm now going to make it. But now, because it was not so firm, then that cassava was from Costa Rica and Colombia. I said, no, perhaps I need to use the cassava, the bitter cassava variety in Uganda that was making better flour and other products. I told my supervisor very fast, and she agreed with me. I came back to Uganda. I went to Western Nile, where that cassava is, and I picked it. Came to Kawanda lab, and I processed it. Now I processed the material from the whole root and then from the peels. Then I quickly ran back to Ireland. Now when I reached there, it was a Saturday. Then I said, Monday I must start. And I told my supervisor, I've brought it. Then he said, so quickly? I said, yes. Then I started Monday in the morning. I went back and went through the same process, and I made what? The polythene sheets. Now, once I made them, now they came out very nice. I was so excited. You know why? Because this was a Ugandan, a local material that we are going to use. In the beginning, I was thinking now, if I get a better cassava from Costa Rica or Colombia, shall we import it into Uganda? But I said, can the breeders now make that better variety? But I said, now I've gotten one from Uganda that can be improved by breeders to make those materials. Now, once I made it, I did not tell my supervisor. So I said, okay, let me wait. So it was a Thursday evening at 5:30 p.m. when others had left. I said, I need to go to the lab, and I was determined to make a cassava bag. Now, I went there, from 5 p.m. to 10 p.m. when the university closes. I did that one. Now, it was surprising. One of our students had bought a watermelon. Remember, I did not tell that person that I was going to use a melon. But I got the watermelon, put it in my cavera while I was returning back to my hall of residence. Now, few meters, 10 meters, it was raining. The paper parted, and the watermelon ran down and split. So, two things. One, I had used someone's watermelon, and then two, the paper bag that I had made, and I knew I was through, then it also parted. Then that night was a nightmare. I didn't sleep properly. I came back to my office in the very early in the morning. I didn't greet people. They were asking me what's happening in Kashub. We need to solve what is worrying you. I said, I'm very okay. Then I sat on my computer. Then I thought that in the evening, I should go back to the lab and do that. Now it was a Friday evening. I said, okay, now I have the evening, but also the weekend when they are away. Now I went back to the lab on Friday evening, 5:30. I did it. I went through the same process. I remembered that I needed to make a cavera that was water resistant. Because I remember that in Uganda, people go and buy things and they put them in cavera and they even go through the rain. So the problem was the rain. So I needed to make the polythene bag that would save me from that. That would be water resistant. So I quickly did it, and I went through and I put the watermelon again, and I moved. I made sure that I put the watermelon of the same size that I used previously. Now I walked a kilometer. I was staying a kilometer away, and it did not part. Do you know? I didn't look at it because I didn't want it to see parting. So I reached in my hall of residence, and it was intact. I was so excited. That night, I was so excited, and even my roommates, my flatmates asked me, what is happening? I said, I've made it. What? I said, I've dropped a biodegradable bag that is going to carry goods. So the following morning, I took it back to the office and also got another sheet like this one and laid it on a seat. I said, okay, I'm going to say. Then I called my supervisor. I said, I have a surprise here. Now, when she came, I said, okay, I've made two things. One, a seat cover for the planes in Europe, and also a biodegradable bag that is going to carry goods. She was so excited. The students I had in the room were so excited, and everybody was so excited. So I felt like I was on top of the world. Even since then, my supervisor decided that she got other students to do research on that caveira, because now she was sure that cassava could make what? Could make caveira. Previously, she was not. She took all what I had done and showed it to the staff, and the staff were so excited. Now, this is the caveira that has a potential to reduce the cost of waste management in Uganda. Imagine removing all the wastes from the channels, from the city, and taking them to Chitezi. And then also managing the Chitezi. How much money is that? Now, this is the caveira that brings about production efficiency. When you are going to produce it, it's cost less than what? Than the other non-biodegradable film. Now, imagine an environment free of caveira. Thank you.
Thank you very much, Dr. Kashyub. It was nice listening to your journey and your story of innovation. If you're watching us from wherever you are, we're entering the last segment of our Science Stories Africa 2020 event. And I'd like to encourage you to follow Dr. Kashyub on his Twitter handle, and also to support his cause. If you're a policymaker, a donor, or someone who knows someone who has money to invest, his technology is worth investing in. Also for Dr. Misaki, the Ebola testing kit is ready for upscaling. If you're a policymaker, a donor, or an investor, this is someone to support. Please reach out to them or to Science Stories Africa, and we will guide you to them. Vicky, do you have anything to say?
Yes, Christine. We would like to thank our audience so much from wherever you're watching from. We say a very big thank you. A very big thank you. We are so grateful. Thank you so much for walking this journey with us. Please, we encourage you, like you see us here at Science Stories Africa, we encourage you to follow the guidelines that your respective governments have put in place to protect you. And we also encourage you to give us feedback about this year's edition. What do you think about it? How should we improve next time? We also encourage you to follow us on our respective social media platforms. You can find us on Twitter, on Facebook, on Instagram, and YouTube. Yes, you can watch all these videos. Please don't hesitate in case you have a great innovator or any researcher you know in your community that is doing great work, amazing research. Please reach out to us. We are available. We shall reach out. You never know. Next year, we might be in your city. So, we thank once again our funders, our sponsors. We can't thank you enough for believing in us, for believing in this idea, and for believing in this dream. That is Cornell, Alliance for Science, as well as Ian Gizad. We thank you so much, and we say bye-bye from us, from Science Stories Africa. It's a bye from us, but coming up next is our entertainers. We have Naava Grey and Davies. I know we've had wonderful performances from them. They're going to give us a last one for the evening. Please enjoy the rest of your evening. Thank you so much.
Bye-bye.
Bye.